Hello and welcome back. This is part 3 of the lecture on cell structure and function. We're in chapter 3. We had just finished up with prokaryotic cell walls. Now we're going to move in a little bit closer into the cell, and we're going to get to the cytoplasmic membrane or the cell membrane. This structure it's got lots of different names. The one thing that it is not is a cell wall. So we'll call it cytoplasmic membrane, cell membrane, phospholipid bilayer. And it's made of lipids, phospholipids, and some proteins. Some of them are integral proteins that go all the way through the membrane, and other are peripheral proteins that kind of hang out on one side or the other of the membrane. Our current understanding of the membrane is that it's fluid, that is, things move around in the membrane. I shouldn't go up and down, like go side to side with each other. Here's a close-up. This is the stuff that I refer to as the Oreo cookie layer, not because it's crunchy, but because it's got that double structure to it. The phosphate heads here in blue, these things are polar or hydrophilic, and the lipid tails, the creamy inside, these bits are hydrophobic. So there's water both inside the cell and outside the cell, and then the creamy insides, those lipid tails, face in towards each other to get away from the water. And that's how there's some organization, let's put it that way. And then sticking through the entire membrane are these integral proteins. So the amino acids that these proteins are made out of, as you might imagine, the amino acids that hang out on the outside, those are hydrophilic amino acids, and the ones that are in the inside, those are hydrophobic amino acids. And so this thing is kind of stable in there because as it kind of slips into the cell, it gets pumped back out. If it slips out of the cell, it gets bumped back into a position. Very clever. All right, what are these membranes for? You've heard this phrase before, selectively permeable. That is, some stuff can pass through with no problem, but then other things, it's like a real barrier to it. Sometimes I think of this like a chain link fence, like air and, well, maybe not butterflies, but bees and flies could definitely get through that pretty easily. But bigger things are going to run up into this fence and they can't get through. People, dogs, that kind of thing. Okay. There's most things are not going to be able to get through this phospholipid bilayer. But because we have built these proteins into the membrane, those allow movement through the membrane. I think of them as like doggy doors. I'll show you what I'm talking about. This helps to maintain concentration gradients. That is, there's more of something on the inside of the cell and more of something else on the outside of the cell. I'll show you. And there are some photosynthetic bacteria that can do kind of photosynthesis using this membrane. Okay, let's talk about this electrical gradient. You know that in your cells, in the phospholipid bilayer, there are these sodium potassium pumps, and they're going all the time. We're using ATP to pay for them all the time to pump sodium out of the cell. It's represented here by these red circles, plus signs in them. These are sodiums. And chloride ions are on the inside of the cells, represented by these green circles with minus signs in them. And there's a whole lot more sodium on the outside than on the inside of the cell. In fact, that's like half the job of the sodium potassium pump is to be pumping sodium out, out, out. This, believe it or not, is hugely important in the functioning of many of our cells. Neurons, muscle cells, etc. They depend on having this gradient set up. Here's an amazing thing. This is a pretty universal constant that if you took a measurement from the inside of the cell and compared it to the outside of the cell, the inside of the cell tends to be about 70 millivolts more negative. So you're more negative on the inside, more positive on the outside, largely because of the sodium ions that you pumped out. Okay, that's true in our cells, and it's true in prokaryotic cells. Amazing. All right, what do I want to tell you about this? Yeah, we work at it and we maintain this difference in we'll talk a little bit about the function of this later. Okay, so here in this cartoon, the blue balls represent the phosphate heads and the lipid tails are in the middle. Here's a protein that goes all the way through. It's an integral protein. The way it's shaped, doesn't that suggest that maybe something can get through here? And we're going to talk about that next. But first, I'm going to ask you a question. Which of the following is a function of the cytoplasmic membrane? It is A, to control the passage of substances into and out of the cell. B, to protect the cell from trauma. C, to produce a capsule for the cell. Or D, to contain the DNA of the cell. And of those options, what we talked about a few seconds ago was controlling the passage of substances into and out of the cell. Right, so in this cartoon, I'm going to represent the passive transport. There's going to be movement of materials, and it's not going to cost anything in terms of energy or ATP. So again, this cartoon, phospholipid bilayers here, very simple. What we mean by diffusion is molecules moving down their concentration gradient. Happens for free. And the kinds of molecules that can move like that through the cell membrane with no problem. One, they better be small, and two, they better not have an electric charge, because this, the lipid part, remember, that's hydrophobic, that forms a barrier. And so the kinds of molecules that can pass through are things that don't have an electric charge. And here's oxygen and O2. Oxygens are sharing electrons with each other. There's no charge. They share the electrons perfectly. So there's no net positive or negative charge. So these can pass right on through. This is also true for carbon dioxide. Trying to think of how much I want to go through this with you. Well, okay, let's put it here. These purple things represent proteins that are embedded in the cell membrane, and they allow passage for certain items. I'm going to look ahead for a second. Yeah, I'm going to get to that next. So I'm going to define these things on the next slide, and then I'm going to show you a version of this picture. It's going to look a little bit different, and I'm doing that on purpose so that you get the sense of like what's important and what is just cartoon. You know, some things are universal. Okay, so let's go on to the next slide with some text on it. These passive processes, once again, what I mean by passive is it does not cost energy, does not need ATP to make it work each time. So the three terms that we're going to use, diffusion is that movement of molecules through the membrane, just straight through the membrane. Facilitated diffusion is the movement of molecules across the membrane, but they need a little help in the form of a tunnel or some kind of opening, like a little window or doggy door, better be there. And that's what the facilitated part is. Like, I need to build something to help this, but it's still for free. I don't have to pay this thing every single time. And then osmosis, it's not my favorite term. It just means it's diffusion or facilitated diffusion, but specifically the molecule that's moving is water. That's what osmosis means. So this is another cartoon, exactly the same principles as what was on the previous cartoon. So diffusion is when you've got a molecule that can pass straight through, and the example I gave you was oxygen or O2. Then facilitated diffusion, we have a couple of examples of this. In one of them, this integral protein is just open. And a number of different kinds of things can pass through. I think of this like a doggy door. Like it's little, it's open, and pretty much anything that size can pass through there. It's not real picky. In contrast, we can have facilitated diffusion that's a little bit more specific. I think of these as doggy doors that only open for the dogs that are wearing the special collar that matches this door. And so not every dog can get through, but any dog that's wearing this collar can get on through. And these are, yeah, they have to be shaped right to bind to this thing so it can pass through. Okay, and then finally, going back to osmosis, it can either be straight up diffusion, just straight through the membrane, or it can be a kind of facilitated diffusion with some kind of aquaporin. These are called. Think doggy door for water. And we have lots of these in our cell membranes to let water through. And so do bacteria. Okay, a little bit more about osmosis, because this is sometimes a kind of a tricky principle to get, that it's possible. So this is a cartoon of a U-shaped tube. It's got water in it and this so-called semi-permeable membrane that can let water through, but can't let these red balls through. And if the red balls could pass through there, the way, diffusion works, that it's going to move from an area of high concentration to an area of low concentration, so the red balls would move across. But this particular semi-permeable membrane does not allow the red balls to pass through. So the only thing that can move is the water. Under those circumstances, then the water is drawn to the side where there's more red balls. And so you end up in a situation where the water's going to, in some ways, being pulled this way, and you end up diluting the red balls on this side. And this, my friends, is the way kidneys work, is that there are these membranes passing material through the cell and getting, say, water to go out. Amazing. It's just what I want you to get from this is the principle that sometimes you've got a membrane, and the other things, the charged particles like sodium or chloride or potassium, they cannot pass through the membrane, but the water might be allowed to pass through, in which case you get these big shifts in fluid, which is hugely important in managing people's health in the hospital. Okay. A little bit more about water movement. The way the water is going to move depends on the concentration of other things like salts or sugars dissolved in the water. So here we have a cell, like an animal cell, and the concentration of solutes on the inside and outside of the cell appears to be the same. And so there is going to be some movement of water, but it's going to be balanced both into the cell and out of the cell. We call this kind of a solution that has the same kind of concentration on the inside and the outside, we call that an isotonic solution. And this is when an IV is put in a patient, you want to make sure that the concentration of that fluid that's going into the body is going to be the same as the concentration of the fluids in the body. We call those isotonic. This is often referred to as normal saline, 0.9% sodium chloride, pretty standard. Why is it so important to match the concentration of the fluids? Because of the next two panels. If very highly concentrated solutes are introduced into the body, that draws water out of the cells and dehydrates the cells. When that solution on the outside of the cell is more concentrated than what's on the inside, this is referred to as a hypertonic. Hyper meaning higher, tonic concentration. And so that causes cells to shrivel down. And then the other situation, if there's solution that's put on the outside of the cell is very much more dilute than what's on the inside of the cell, it is possible for the solutes on the inside of the cell to draw so much water into the cell that the cell swells tremendously. In extreme cases, it can cause the cells to lyse, to burst open. Okay, so you'll learn more about that once you're in nursing school and talking about, different kinds of IV fluids. All right, now let's see what's going on down in these lower panels, because we're putting them in the same we're putting this cell that has a cell membrane, but also has the cell wall, we're putting it in the same three kinds of solutions. In the isotonic solution, kind of looks the same, fluids moving into and out of the cell, no big deal. In the second one, where we put the cell in a hypertonic solution, a very concentrated solution, that does cause the cell to shrivel down, to dehydrate. But on the outside of the cell wall is so rigid, it kind of looks the same. And then finally, here's the big secret to how fabulous cell walls are for these bacteria. When the cell is placed into a hypotonic solution, a very dilute solution, there's going to be some pressure for water to be pulled into the cell. But because of the rigidity of the cell wall, that's not going to be lethal for this cell. That cell's going to resist lysing. And so that's one big advantage that bacteria have over, for example, animal cells. They're more resistant to lysing in a hypotonic solution. And again, the summary of, a couple of things. So when we talk about those movements, we're talking about hypotonic and hypertonic solutions, in which way the water moves. What we're talking about is osmosis. The movement of these water molecules, either directly through the membrane, which happens a little bit, or through these pores, aquaporins, or transport molecules that allow water to pass through. Okay, so that was all passive transport. Now we're going to shift gears a bit and look at what we refer to as active processes or active transport. This means we're going to pass molecules through the membrane again, but this time it's going to cost us energy. We're going to have to pay for it, usually with ATP. And here's the spoiler alert. When you do active transport, the reason you have to pay for it is because you're shoving molecules against their concentration gradient. You're moving them to an area where there's already a lot of that molecule and you're trying to move more of it in there. That's going to cost you. Okay, and then I'll very briefly talk about group translocation. Okay, so active transport. Again, this cartoon, blue is the phosphate heads, yellow is the lipid tails. In purple, we're representing the proteins here embedded in the cell membrane. And, excuse me, uniport just means I'm moving one thing in one direction. I'm trying to get a molecule, this black molecule, whatever it is, from the outside of the cell to the inside of the cell. But I already have a bunch on the inside of the cell. Well, it doesn't matter, I want more. So I'm going to pull it in, and that's going to cost me because this is going against the concentration gradient. So I better pay for it with ATP that causes this protein to change shape and allows this molecule to come into the cell against its concentration gradient. That's uniport. With antiport, we're going in opposite directions. An example of this would be, for example, the sodium-potassium pump. We'd be pumping, let's say these squares represent sodium, pumping sodium out and pumping potassium in. And again, that's going to cost us something because we already have a bunch of these blue squares on the outside and we're wanting to pump more out. And we already have gray balls on the inside and we want to pump more in. So that's antiport. This one, I don't know how much we're going to talk about this semester, so I may not spend a lot of time on it. In a nutshell, there's a uniport system like this one, but this time I'm pushing this molecule out and I am letting it take the ride back in. Well, let me rephrase that. This thing is now going to be moving down its concentration gradient, and it brings a buddy along for the ride. There are mechanisms in the kidney, for example, that work this way. We can talk about that another time. Again, this is another cartoon representing the same thing. Uniport, I'm moving things in one direction, but I got to pay for it every time. Antiport, I'm moving two different things in opposite directions, also paying for it every time. The example I gave you here was sodium potassium pump. An example I can give you for Uniport would be a calcium ion channel that I'm having to pay for. And Symport, I'm moving one molecule, I'm paying for the one molecule, and then I'm letting the second one kind of take a ride. I'll show you an example if we come to it later this semester. And then that final example of active transport is group translocation. It basically means I have a molecule on one side of the membrane. I want to get it across the membrane, but as I get it across the membrane, I'm going to put a chemical group onto it. Like, for example, this phosphate group. I'm going to attach that to this glucose. And so this becomes a glucose 6-phosphate on its way in. And I have one question for you about this. Active transport across the cytoplasmic membrane differs from facilitated diffusion in what respect? A, active transport does not require energy. B, active transport is used to move materials against their electrochemical gradient. C, active transport is used to move materials with or down their electrochemical gradient. Or D, only active transport uses transport proteins. And let's go through these one at a time. Active transport does not require energy. False. Active because we're paying for it. We're using ATP every time. Active transport is used to move materials against their electrochemical gradient. I think I never mentioned the electro part of this. I talked about the concentration gradient. Yeah, so you are moving things against their concentration gradient or electrochemical gradient. So B is correct. I want to go through the other two though. Active transport is used to move materials down their electrochemical. If you're moving down the gradient, it doesn't cost very much. It's like, it's easy. It's like going down a slide, you know, it's easy. That would be passive transport. And if it needs a protein to allow the thing to move through, like for example, water needs that transport or aquaporin protein, that can happen, but if I don't have to pay it every time, that's not active transport, that's facilitated diffusion. And then that's where this one comes in. Only active transport uses transport proteins. That's not true. Facilitated diffusion also needs transport protein. The difference is, do you have to pay the transport protein every time? And if you have to pay the transport protein every time, that's active transport. If you don't, if you just built the protein and put it in place, but you don't have to pay it every time, that's diffusion. That's facilitated diffusion. Okay, and that's the big difference. All right, that's the end of part 3. I'll see you over in part 4.